So you ready, guys? All right, so let's go ahead and get into this. We're going to start off with Chapter 9. Chapter 9 deals with microbial genetics. So one of the big concepts we talked about in this chapter is the difference between genotype and phenotype. Okay? Remember, genotype are all of the genes found within an organism, and the phenotype are the genes that are actually expressed and used by that organism. Okay? And remember, these are often different. Okay? Most of the time, an organism is not going to use all of its genes. Okay? It's only going to use a fraction. So genetic material, or DNA, has to be able to self-replicate, and then when it does this, it needs to be accurate. Okay, so there should be little error in this process. All right, so the structure of genetic material. Does anyone remember what a genome is?
DNA.
DNA, all the DNA, right, inside of the cell. What about the chromosome? Or a chromosome?
Segments.
Yep, so it's segments of the DNA, right? So some organisms will have multiple chromosomes. All of those chromosomes equal the genome, okay? And then what about a gene?
All the thousands of chromosomes.
So it's a region in the chromosome that codes for something, right? So a portion of the chromosome that codes for something the cell can use. Whether that's a protein, in case of a structural gene, RNA for RNA machinery, or regulation, so on-off kind of switch. So this is a good slide, slide 8. It's a nice summary between the differences between eukaryotes and prokaryotes. Okay, we've talked a lot about this throughout the semester, and it's going to keep coming back. Okay. So remember, eukaryotes have multiple chromosomes, and they are linear, okay, in structure. Where bacteria or prokaryotes have usually one chromosome, and it is circular in structure. Eukaryotes can also come in diploid, which essentially means they have two sets of chromosomes. Okay? One from mom's side, one from dad. All right, so DNA itself is made up of nucleotides. Okay, so these are the individual building blocks of the DNA molecule. Okay, and remember, the only thing that changes from one nucleotide to another is the nitrogenous base. There's 4 of them. Does anyone remember what those are? Adenine, thymine, guanine, and cytosine. Right, A, T, C, and G. Okay, good. And remember, DNA is a polymer, so it is a chain of these nucleic acids. Okay, these nitrogenous bases. And remember, this chain or polymer often sticks to another chain, making a double-stranded molecule. And remember that these nitrogenous bases stick together in a certain way. A's with T's and C's with G's. Okay? Okay, DNA replication. All right. Now I'm not going to expect you to go through all the steps involved in this. Okay, but I do expect you to know these 5 enzymes. Okay, the first 5 in this table. So we'll start off with helicase. Does anyone remember what helicase does? It unzips the DNA, right? So it takes the double-stranded molecule and turns it into 2 single-stranded molecules. Then we have primase. What does primase do? Adds RNA primers. And what are those RNA primers there for? Right, it tells the next enzyme where to start. Okay? The next enzyme, DNA polymerase 3, binds to those RNA primers, right? And then what does it do? What is its function? It does proofread, that's one of its functions, but its primary function is to add in nucleotides, right? Creating a double-stranded molecule. So it takes a single-stranded molecule, adds in complementary bases until it becomes double-stranded. And if it does that on both sides, you get 2 double-stranded molecules. Then DNA polymerase 1 comes in. What does polymerase 1 do? It removes the RNA, replaces it with DNA, because that's what we want, DNA, right? And then ligase, what does ligase do? Fills in any gaps, makes the DNA really strong, okay? So you don't need to know every single step, but you need to have a working definition of what each of these enzymes do. All right, so DNA replication, we just talked about that. When does DNA replication occur within the cell? Is it duplicating its DNA all the time just for fun? No. Only when it's dividing, okay? So duplication of DNA only occurs when cells are dividing. When cells aren't dividing, what is DNA there for? What is its main purpose? Right, it's the template, master template or master copy for protein production, okay? So DNA's primary purpose is to eventually be converted into protein, okay? So remember, this is known as the central dogma of biology. So if I have a cell and it needs to make a protein, it would start at the DNA, it would then transcribe that into RNA, and then that RNA would be translated into a protein, okay? That's the basic process of how this works. This is a nice little cartoon diagram describing this. So DNA gets transcribed into RNA. There's many flavors of RNA. This then gets translated into a protein. Okay? Where does translation occur if you're a eukaryote? Where are the ribosomes in eukaryotes? Outside of the nucleus, right? In the endoplasmic reticulum or in the cytoplasm. Okay? So translation in eukaryotes occurs within the cytoplasm, okay, or outside of the nucleus. What about in prokaryotes? Where does it occur? Anywhere, okay? They have no nucleus, so they just float around all over the place, okay? So it's one easy way to tell if you're dealing with a prokaryote or a eukaryote. It's in the nucleus, or if it's outside of the nucleus, then it's in a eukaryote. If it's anywhere, then it's in a prokaryote. And remember that 3 consecutive nucleotides in both DNA and RNA will eventually code for one single amino acid. Okay? This is known as the triplet code. So it's 1, 2, 3 nucleotides, that's one amino acid. Okay? In DNA, these 3 consecutive nucleotides are called triplets. In the RNA, they're called codons. And again, this is converted into one single amino acid. So a little bit about RNA. What's the major differences between DNA and RNA? Uracil instead of thymine, right? So you have a U instead of a T. Okay, that's one difference. What's another difference?
Single-stranded.
It's often single-stranded, right? And then the other really big difference, it's in the name. It's less stable, but why is it called RNA instead of? It's got a ribose sugar instead of a deoxyribose sugar. Okay, so those are the 3 major differences. And remember, there's 3 flavors of RNA. Messenger RNA. What does this do?
Codes for protein.
It codes for the protein, right? This is where you would find those triplet codes, those 3 consecutive nucleotides. The next one is transfer RNA. What does transfer RNA do?
Translates it into amino acids.
Translates it into amino acids. This is the guy that kind of figures out, okay, 3 consecutive nucleotides, that means this amino acid. Okay? And then the ribosomal RNA or rRNA, what is this used for? Making the ribosome, okay? If you don't make the ribosome out of RNA, then it would never be made because it makes the proteins, right? Oops. Transcription. So transcription is relatively simple. One enzyme, RNA polymerase, binds to a gene, the promoter. It makes a copy of that gene into RNA. Okay, very, very straightforward. Where would this happen if you are a eukaryote?
Nucleus.
In the nucleus, right? Because you need the DNA. Where's the DNA? In the nucleus. What about if you're a prokaryote? Where does this happen?
Anywhere.
Anywhere, right? Because there's no nucleus. Then on to translation. So translation, this is where we convert our RNA into amino acids, okay? And when you have a chain of amino acids, you get a protein, okay? It gets folded and all that good stuff. Now, again, I don't expect you to know the ins and outs, all the details associated with translation. Okay? What I do expect you to know is that it occurs in the cytoplasm for both eukaryotes and prokaryotes. And it's where the mRNA binds in the ribosome, okay, opening up the codons and allowing tRNA to bind and bring with them amino acids. Okay? And when this happens throughout the entire chain of the mRNA string, you get a protein. What does it mean to have redundancy in this genetic code? Right, so there's multiple codons, okay, for each amino acid. Why is it this way? In case there's a mistake, it's less likely to cause a problem, right? Because you might get the same exact amino acid. Good. What type of organism do you think this is occurring in? Eukaryote or prokaryote? Why are you saying that? Do you see a nucleus?
I can't see it.
You probably get some glasses.
I'm about to use that.
I can't see it from here, but
I had a girl come and take an exam once, and she thought she was using eye drops, but in reality it was something that dilates your pupils.
Oh.
And so she came in and she couldn't see the exam, so I put it on the board for her, and she sat back there and like looked at us, and she saw it.
I think you should just give us like points for us today by doing oral.
Well, then you'd have to get rid of all your notes. And I would have to just ask you random questions with nothing
Who
to
just has those?
Yeah, it's probably a little worse to do an oral exam. Okay, so this is a prokaryote because there is no nucleus. You've got DNA, RNA, and then translation, all happening at the same place. Okay? DNA is being transcribed into RNA, and then that RNA is being translated into protein. In a eukaryote, this would not happen at the same place because there would be a nucleus right here. Okay? These guys would be outside of it, and the RNA would have to leave the nucleus to be translated.
So in sorry, but
Uh-huh.
in eukaryote, everything happens in the same place in the beginning. Is that DNA replication?
So DNA replication always occurs in the nucleus for eukaryotes, okay? And transcription occurs in the nucleus for eukaryotes.
Okay.
Translation occurs outside, in the cytoplasm. All right. Now, not only does translation have to occur outside of the nucleus, so that RNA has to leave the nucleus. Remember, there's also a lot of garbage RNA inside, which we call introns, and these have to be spliced or removed before they can be translated into protein. Okay? So in eukaryotes, there's two problems. RNA has to leave the nucleus, and also you have to splice out all of the garbage, okay, or these introns. Remember, this intronic RNA is useless, so it needs to be cut out, and then you're left with just the good stuff, or the exon. Okay, let's talk about what happens with problems in DNA replication. Okay? When you have a problem in DNA replication, you get a mutation. This is a change in the nucleotide sequence, okay, or the nucleotides of that DNA. When this occurs, we call the organism a mutant. And the organism that was the original guy that didn't have those mutations is known as the wild type. Okay? And remember, these can be either spontaneous or induced. What is a spontaneous mutation? How would a spontaneous mutation occur?
Just happens.
Just happens. Shit happens, right? It just, some weirdness happened during replication, and you're stuck with a mutation. Okay? What about an induced mutation?
Brought on by something else.
Brought on by something called a mutagen. Okay? This is a chemical or physical energy that screws up the DNA. Okay? And as far as mutations are concerned, there's two major groups, point and frame shift. Okay? What is a point mutation?
It adds in.
Or deletes it.
It's going to modify one nucleotide at a time. Okay, so a single change in one nucleotide.
So the change can either be added to or deleted from?
It generally is just changing it from an A to a T or a C to a G, something like that, right? 'Cause if you take it away, you can then develop a frame shift mutation if it's in the wrong spot. Okay? So it's just changing the nucleotide sequence. What about a frame shift mutation?
It adds in multiple nucleotides.
Right, so you're adding in or taking away multiple nucleotides. Remember, when this happens, it changes the reading frame, okay? The reading frame is basically how the protein is made. The ribosome goes, every 3 nucleotides, I'm going to get an amino acid, okay? So if you go in and you take away 5 of those amino or nucleotides, then it's going to screw up everything downstream of it, because now it's just going to go to the next 3 nucleotides and look for an amino acid there, okay? This results in a completely different protein and a failure. So which one do you think is going to be the most severe? Point or frame shift? Frame shift, right? Now there are point mutations that are severe, like a nonsense mutation, where you change it so that it becomes a stop codon. This causes big problems. But most point mutations aren't going to be that serious. Alright, so mutations in general are usually harmful to the organism. Okay, they're mostly negative. However, there are cases where mutations can be positive. And when this occurs, the organism gets a growth advantage, and this allows for natural selection and evolution to occur. Okay? So it's these small mutations or small changes that are beneficial. Again, this is the minority. It happens not that often, but when it does happen, this allows for organisms to change and evolve. All right, so the last thing we're going to talk about in this chapter is recombination. What is recombination?
When an organism is having to divide. Or
No.
without, I don't know, I can't read my own.
So recombination is where bacteria pick up DNA from somewhere else. Okay, usually another bacterium, but occasionally it can even be from the environment. Okay, and this can occur in 3 ways: conjugation, transformation, and transduction. What is conjugation?
Bacterial sex.
Exactly, bacterial sex. You get bacteria, they get next to each other, and they shoot across DNA. Okay? So it's thought of as bacterial sex. What about transformation? What is a transformation?
Taking up DNA from dead bacteria.
Taking up DNA from dead bacteria in the environment, right? It's floating around, you just go up to it, grab it, take it in, boom. And what about transduction?
Infection from viruses.
Right, so bacteria gets infected by a virus. This virus carries with it some sort of DNA in which it then can utilize. I thought I was going to sneak one. That wasn't. Oh. Okay. That is it for chapter 9. On to chapter 11. Chapter 10 was mostly laboratory techniques, so that would be something we would see in the practical, okay? And as I said, most of chapter 10 was really just for your own information. A couple things, though, I do expect you to know for the practical, like PCR and things. But for this exam, it's not pertinent. So, 11, physical and chemical control of microbes. This chapter has a lot of definitions, okay? A good rule of thumb for any of these is look at the bold words and know what they mean. In case you haven't noticed, those are very helpful for me when I'm determining the matching questions. So, sterilization. What's sterilization? Kill all organism, right? What about disinfection?
Destroy most.
Kill most organism, right? 99.9%. What about antisepsis? Disinfection on tissue, right? On a living thing. And then decontamination?
Getting rid of crap.
Yeah, so physically removing it, not necessarily killing it, just getting rid of it. Okay? And remember, organisms have different resistance mechanisms to some of these ways of killing them, okay? Generally speaking, most of the organisms we deal with are going to be least resistant, which means they're very easy to kill, okay? However, there are high resistance organisms like endospores and prions. Those are the ones that we really need to bring out the big guns for. So again, sterilization is to remove all living things. Disinfection is to remove most living things. By far and large, this is what we do more often. Okay? Antisepsis is removing most living things on living tissue. And remember, if you see a word with "side" at the end, it tells that it's going to kill the organism, or tells you it's going to kill the organism. Where if you see stasis or static, it implies that it is only going to slow the growth of the organism, okay? Prevent division. And then decontamination, this is physically removing the organism. And if you do this with a chemical, okay, that also can weaken the organism, this is known as sanitation. Okay? This is all common sense, right? Everyone knows what death means, right? Organism dies, no longer can divide, goes away. So how do these agents work? Okay, there's 4 places you can target: the cell wall, the membrane, protein nucleic acid synthesis, or protein function. What happens when you target the cell wall with these chemicals? What happens to the organism? You break the cell wall down, and then the organism explodes, right? It lyses. Good. What if I break down the cell membrane? What happens to the organism?
The phospholipids can be broken.
So you break down the phospholipid bilayer, and then the cell no longer can control its environment. Okay. What about protein and nucleic acid synthesis? What happens if you just stop this? This is a no-brainer. They die. No protein, no DNA, no RNA, you die. You can't make anything. And then protein function. What happens when you disrupt the protein function? Okay, or denature the protein? The cell gums up into a big mess. Okay? When you denature proteins, they all lock together, and this becomes kind of like snot inside of the cell. Alright, so now let's get into physical control. Within physical control, it's mostly using either heat or radiation. Okay? We'll start off with heat, and you can use either moist or dry heat. Which one is more effective and efficient? Moist, right? Because you're using energized water molecules that help break the organism up. Dry heat is primarily only used when you're dealing with something that's very sensitive and can be exposed to moisture. Alright, so the most common form of sterilization is steam under pressure. What is this often called? Autoclaving, right? This is often used because it is the most efficient, at least energy-wise. You heat it up to 121 degrees Celsius, pump in some steam at 15 psi, and let it go for 30 minutes. Okay? Just keep in mind that this doesn't work for every instrument because it does have to be able to withstand high temperature and high pressure. You can also use non-pressurized steam, but this generally is not a good way of sterilization. This is a form of disinfection. It's going to kill most organisms, but not all. Pasteurization. This is where you heat a liquid to a temperature, usually relatively low temperature, for a short period of time until most organisms are destroyed. Okay? So this is, again, another form of disinfection. It is a way of removing most organisms from a liquid without changing the liquid too much. Boiling water, again, another form of disinfection. It's really not that efficient. And then dry heat, relatively inefficient. The one place this gets used a lot is in the lab by incineration. Okay, so burning the organism alive. What about cold? Does cold work well as a disinfection or sterilization technique? No. Why? Just slows it down, right? So what would you call this? A static approach or a cidal approach? Static, right? So it's stopping growth or slowing growth, but it is not destroying the organism. In fact, it's actually preserving the organism. The other form of physical control is radiation. We've got ionizing and non-ionizing. Which one's more potent? Ionizing, okay? This is where electrons are emitted. It can penetrate through most substances. And when it does, it gets into the cell and shears or cleaves the DNA into pieces. Okay? Where would you use ionizing radiation? This is often used to cold sterilize. Okay? So this is where you would sterilize something, but without generating any heat. So it gets used a lot with like sensitive plastics or food items and things like that. Non-ionizing radiation is generally considered to be weaker, and that's primarily because it does not penetrate. Okay? It's UV light, so it can only attack cells that are exposed to this light. It works by creating pyrimidine dimers or fusing nucleotides together. This causes errors and eventual cell death. And where does non-ionizing radiation get used? Anyone remember? Primarily? Water treatment. Okay? Because water is clear, so the UV light can penetrate it very easily. Alright, so decontamination, this is where you physically remove the organism. On a solid surface, this is pretty self-explanatory, it's just scraping it off. In liquids or gases, this is primarily done through filtration. This is where you pass a liquid or your gas through a membrane that has little holes or pores in it. This blocks the organism from going through it. Okay? And so you're left with a clean liquid or clean gas. What's the major drawback to using a filter? Viruses get through, right? So it doesn't work well for all pathogens. Viruses make it through no problem. All right, on to chemical agents. Okay? For chemical agents, all of these are what we call reactive. That means they tend to have a free radical or a free group associated with them that can pull apart and dismember proteins within the cell. Okay? So most of these function by disrupting the cell functions, primarily by disrupting proteins and enzymes. So the first group of chemicals are halogens. Okay? Halogens are chemicals that have a halogen group associated with them. The two most common are going to be chlorine or iodine. Okay? These again work by disrupting protein function. Okay? Most of these chemicals are going to work in that way. Phenols. So phenols have a phenyl ring with a hydroxyl group. And these work primarily again by disrupting not only proteins but also cell membranes, okay, and cell walls. However, these tend to only work at high concentrations, which are often poisonous to humans, so we don't use them terribly often, at least not anymore. If you combine a phenol with a chlorine molecule, you get chlorhexidine. Okay? This is getting the best of both worlds. You get the benefits of the reactive chlorine group and also the membrane permeability of the phenol or phenol groups. Alcohols have hydroxyl groups. These work by disrupting protein but also desiccating the cell, okay, or removing the water from the cell. Peroxides have very reactive oxygens, so free oxygens, that literally go in and just destroy anything they come into contact with. Okay? Oxygen is a very reactive molecule. But remember that a lot of organisms have enzymes called catalases that can break up these hydrogen peroxides. So they're not always effective. Detergents or surfactants, these work by disrupting the membrane. Okay? So this is one exception to what I said. Most of these other chemicals are going to disrupt protein. This guy does not. It disrupts the membrane. When the membrane is disrupted, the cell can no longer control its environment, and then it quickly dies. Metals, again, these disrupt proteins, especially enzymatic function, but they're also toxic to humans. Then aldehydes. Aldehydes have two, or have an aldehyde group associated with them, and most of the more effective ones are going to have two of these. And these guys work by cross-linking protein. So instead of disrupting the protein, they stick them all together, and this causes the cell to die. Okay. That's it for chapter 11. Chapter 12. Chemotherapy. Okay? So chemotherapy is using a chemical to treat a disease. For our purposes, this is going to be an infectious disease. Okay? And remember, when you are treating someone with a chemical, you want to get rid of the disease-causing agent, in this case a microbe, but leave the individual cells alone. Okay? Does anyone remember what this concept is called? Where you're targeting only the bad guys, the pathogens, and not your own cells.
Antimicrobial chemotherapy?
That's the general name of it, but the process in which you're just targeting the bad guys and not your own cells. It's got a name. I think it's toxicity is part of it. There's another word in front of it.
Cytotoxicity?
No, that's usually bad. Selective cytotoxicity. Very selective toxicity. So you're selectively targeting only the bad guys, not the good guys. This is a nice slide describing, a bunch of definitions. Okay, most of these I'm sure you've heard of before. So chemotherapeutic drug. This is any drug used to treat, or any chemical used to treat a disease. Prophylaxis is administering a drug before infection or disease takes hold, in hopes of preventing it. Antimicrobial chemotherapy is chemotherapy targeted at microbes. Antimicrobials are any compounds that destroy microorganisms. Antibiotics are compounds that destroy microorganisms and are natural in origin, so they come from nature. Semi-synthetic drugs are antibiotics that have been modified in the lab. And synthetic drugs are completely designed in-house, okay, or in a chemistry lab. And drugs can be either narrow spectrum or broad spectrum. Narrow spectrum means they target a specific type of microorganism, where broad spectrum can target large swaths or groups of microorganisms. So in case you're wondering where I got the selective toxicity from, it was from the slide. Just thinking ahead, turn to busy guys. Alright, so this is a nice summary slide of the 5 major groups of drugs that we talked about. Okay? Now for this exam, I don't expect you to really know any of these drugs or particular drugs by name or by heart. Okay? Instead, what I really expect you to know is the 5 groups, how these drugs work within each group, generally speaking, and the pluses and minuses of each. Okay? So let's start off with the cell wall inhibitors. These cell wall inhibitors inhibit the production of the cell wall. Does anyone remember what they primarily do to the cell wall? They don't really break it down per se, they prevent it from being produced, okay? And primarily by preventing cross-linking. So remember, peptidoglycan has a bunch of strings. The only way it makes a cell is if it's sealed up, okay, by cross-linking. These drugs tend to prevent this, so there's strings left, and that's it, and the cell explodes. The benefit of these drugs is they work relatively well, okay, against most organisms with one exception. So anyone remember the type of organism that these tend not to work very well with? Gram-negative organisms, right? Because remember, gram-negative organisms have a thin cell wall, which they don't rely on too heavily, okay? So these drugs tend not to hurt them. Cell membrane drugs. So cell membrane drugs disrupt the membrane. Again, you disrupt the membrane, the cell loses its control over its environment, and then it quickly dies. The benefit of these drugs is they work. What's the drawback to these drugs? They're not very selective, right? So if you were to ingest them or inject them, it would probably cause a lot of cell death. What about DNA or RNA synthesis inhibitors? Okay, these prevent DNA or RNA from being produced, so it sounds like a great idea. But what's the major drawback to these drugs? cell death. They're not very selective either, okay? And the main reason for that is that the cell mechanisms involved in making DNA and RNA are virtually the same in eukaryotes and prokaryotes, okay? So they often target our own cell bits and pieces. Next, we have protein synthesis inhibitors. What do these drugs target? The ribosomes, right? And why can we target prokaryotic ribosomes? They're different than eukaryotes, right? They go about making protein in a very different way. Okay? This is why this is the largest class or group of drugs that we have. Okay? So these are often very nice drugs. They work well against most organisms, and they have low toxicity in humans for the most part. Then lastly, the folic acid synthesis inhibitors. What do these guys do? They block the pathway to produce folic acid. Why does this not hurt us? Do we make folic acid? No, right? We steal it. So the fact that we don't make it, we can target it in these guys who make it and require that to occur. Okay? And remember, folic acid drugs tend to, organisms tend to evolve and develop resistance to them very quickly. So these are rarely administered on their own. They're usually given in combination with other therapies. So those are the big take-home messages. Now, out of these groups, there's two big ones: cell wall inhibitors and protein synthesis inhibitors. I don't expect you to know all the different drugs in each one of these groups, even though we talked about them. What I do expect you to have, though, in your mind, is one drug class from each of these groups and a little bit about it and how it works. Okay? So for cell wall inhibitors, a good one would be penicillin. Okay? Penicillins is probably the most well-described antibiotic around. And again, these inhibit the cross-linking of peptidoglycan. By doing that, the cell then explodes. For protein synthesis inhibitors, you can really pick any, maybe tetracycline. Tetracycline binds to the A site of the EPA site in the ribosome. This inhibits translation. And again, this is a reversible process, so if you remove the tetracyclines, the cell can rebound and survive. Okay? Also, these are very broad spectrum drugs, which means they target many, many different types of organisms. All right, so that's most of this chapter.
So you said we have to know specifically what one antibiotic does?
In each of the groups. So if I were to ask you a question about it, I would say, give me an example of one cell wall inhibitor and how it functions. Okay?
Do you know what that specific one does, or what they just do in general? What that
One particular one from each of those groups. Right.
And just give me a short answer. From the cell wall inhibitors and from the protein synthesis.
Right, because those are the two big ones. Right. And we talked, through the lecture, we talked about each of these. All right, so enough of bacteria. Now let's move on to fungal organisms. Generally speaking, it's harder to treat fungal infections than bacterial infections. Why is that? What makes fungi different? Eukaryotic, right? So they do a lot of the same stuff we do. Okay? So it's difficult to specifically target them alone. However, there are two places you can target. Okay? Anyone want to guess at one of them? It's one of those unique molecules that I made you know for the first exam that fungi have.
Oh,
Chitin and another big one?
oh, ergosterol.
Eugesterol, right? So remember, eugesterol is a sterol found in the membrane of fungal organisms. This is found only in fungi. So this is actually this is the easiest target for us to go after with antifungal drugs. The two classes of drugs that target eugesterol are the polines, which bind to eugesterol and disrupt the membrane, but they're very toxic to humans. And the azoles, okay? The azoles target the production of eugesterol. So these tend to be less toxic because they're going a little bit upstream. Okay? A little hint, if you see a word with drug with the ending azole, that tells you it is an azole, okay? Which is an antifungal drug. Antihelminthic drugs. So there's only a handful of these available. Why is that true? Why don't we have hundreds of these types of drugs? Pretty cheap.
Since they're bigger organisms, they don't have resistance to drugs.
Right, they're bigger, larger, multicellular eukaryotes. So they don't mutate and evolve often, okay? So that means we don't have to have many drugs against them. And remember that a lot of times these drugs only work against certain portions of the life cycle, usually larvae. So if it's only targeting the larva, how long would you have to take these drugs for? Years until the adults die of natural causes. Because if you stop taking the drug, then the adults just start pumping out more babies, and then you get sick again.
Would you take two types of drugs? One that targets the larva, and one that kills the adults?
Usually so the 3 drugs that get used a lot work in very similar manners. So usually, no. You just gotta wait it out, or pull the worms out. There's lymphatic worms that can get like meters in length, and you just pull them out of your lymph nodes. Ever hear of elephantitis?
Yeah.
That's what causes it. So there's a filarial parasite that gets in your lymphatic system, and you just build up fluid. And so they'll take a stick, they'll cut a little hole, pull the organism, and you just roll it out slowly. All right, enough of that. Antivirals. What do antivirals do?
Block virus from getting into the cell.
Right, so they stop the viral life cycle, right? And imagine this is relatively difficult to do because viruses take over cells and use cell machinery. So at least from a chemical point of view, it can be hard to tell one from another. But the 3 easiest ways to do this is to prevent the virus from getting in, stop the viral life cycle by stopping transcription and translation of the virus, or stop the maturation or the production of viral particles that can then infect a new cell. And out of these antivirals, one of the most important is acyclovir. This is the first antiviral described in the 70s. And again, it targets herpes viruses. Okay? It's very potent, and it's also a prodrug, which means it has very little side effects because it only becomes active in a cell that is virally infected. Interferon, we'll talk about that later. All right, so what is drug resistance?
When the body's immune to the drug itself.
See, a lot of people tend to make that mistake. You got to be careful because it has nothing to do really with the body per se.
The cells itself.
This cell, the microorganism's ability. Okay. Drug resistance is where you have an organism that can survive in the presence of a drug that at one point killed it. Okay. And again, it really has absolutely nothing to do with the human body. Okay. If a drug is eliminated by your body too fast, then it can look like it's drug resistance, but that is a concept called tolerance. Okay, and it's a very different concept. So, drug resistance, and organisms do this. How? How do they develop resistance to a drug? By mutations and evolution, right? So natural selection, which is selective pressure the drug puts on the organism. This then creates a good environment for organisms that mutate in such a way that they then can survive in the presence of the drug. Once they can survive, then they can take over the population because no one else can live there. They then become the dominant form, and now you have a new mutated species that can survive in the presence of that particular drug.
Isn't that what you said happens with HIV?
Yep. It happens with viruses, bacteria, everything. The problem with HIV is that HIV does it very, very quickly, in weeks. Okay, that's how quickly it can evolve. So that's why you go off your drugs for a couple weeks, and then try to take your drugs again. They stop working because the virus has evolved.
So how does HIV working? Isn't there like a pill you can take if you how does that work then?
So there are drugs available, right? I think the ones you're talking about are PrEP, the pre-exposure prophylaxis. And what that does is it stops the viral life cycle very early on. So for the virus to evolve in such a way that it can get away from drugs, you already need to be infected so that it can amplify in your cells and kind of spread very quickly. But if you take PrEP, it blocks the virus from getting in altogether, and then you have nothing to worry about. But there are HIV viruses out there that can survive in the presence of PrEP, so that's why PrEP isn't 100% guaranteed. So how do organisms develop resistance, and then what do they actually do to allow for this to occur? Well, they basically can either block the drug from getting in or change where the drug binds, okay, so that it no longer functions as it should. It's either blocking it, getting it out of the cell, or preventing it from getting in the cell, or blocking where it binds so that it can no longer do its job. This is just a nice little cartoon describing how a resistance can develop pretty quickly. So here you have two organisms that have spontaneously mutated in such a way that they can survive in the presence of this drug. Everyone else that is gray does not have this mutation, so then they quickly die. What you're left with are just these two guys who are alive, and now they get all the plate to themselves, they get all the food and nutrients. They then over time can grow and divide until they take over the whole plate, and now they are the dominant form, okay, because they're the only ones that can survive on that plate. This is a nice summary slide, so 55, of how we can prevent drug resistance. Okay? First and foremost, we can limit the drug use. How would this prevent drug resistance? So you limit the exposure, right? So if you limit the drug use, you're limiting the amount of organisms that are exposed to it, and so then they won't develop resistance to it. Okay? What about proper drug use? Taking your drugs until they're completed. How does this prevent drug resistance? Right, well, yeah, that. And also, if you take the drugs for the entire time, you're going to kill the stronger guys, or the ones that were harder to kill, right? So if you take a drug for a couple days, you kill 99%, you still got that 1% left. So you want to keep taking your drugs to make sure that they go away as well. Narrow-range antibiotics, so target only the organisms that are causing the problem. How would this benefit or reduce drug resistance? Well, if you take a narrow-range antibiotic, it's less likely to cause complications in your patient, which means they'll take the drug, okay? Reducing the amount of resistance. And then multiple drug treatments, so where you're taking multiple drugs, targeting different portions of the organism's life cycle. And why would this be beneficial? Well, it's pretty difficult, if not impossible, for an organism to develop resistance to 3 different areas or different portions of the cell. So by taking multiple drugs, it greatly reduces the likelihood of this resistance development. So how do we determine what drugs to use and at what concentration? So you guys have done a little bit of this already. The cheap and cheerful method is something called the diffusion assay, or the Kirby-Bauer test. This is where we streak our organism on a plate as a lawn, add antibiotic discs, and then measure the zone of inhibition, or the clearing of organism around that disc. Okay? The larger the zone of inhibition, the more susceptible the organism is to the drug. The smaller the zone of inhibition, the more resistant the organism is to the drug, because it can grow in very high concentrations of that drug. And if we want to know more about concentrations in numbers, then we would use an E-test or a dilution assay, and this allows us to determine the MIC, or minimum inhibitory concentration. This is the lowest effective dose of that drug. And this is used to come up with the therapeutic index, or the TI. The TI, or therapeutic index, basically tells you a ratio between the toxic dose in a human and the effective dose. The larger the number, the better the drug. Because now you have a big difference between the toxic and effective dose, meaning it's less likely to cause side effects. All right, on to chapter 13. So chapter 13, again, has a lot of definitions. Anything that's bold is probably an important thing for you to know about. So infection, this is being colonized by an organism that may potentially be pathogenic. Okay? A pathogen, or pathological state, is where you're infected with an organism that is disrupting your cells and tissues and causing disease. And an infectious disease is when you have an organism that is causing disease in the body. Okay? So resident biota, what are your resident biota? Resident microorganisms. Yeah, they're the guys that are in you, right? So we have tons of organisms in us, primarily in our gut, and these are the normal organisms that are present in our body. Okay? Remember, these make you healthy. Without them, you would not feel very well. And so it's important that you have these resident organisms. Most of these are going to be found in the intestinal tract, but really anything that is exposed to the environment could potentially have microorganisms. And these can be residents, which means they stick around for long periods of time, change very slowly. It can be transients, which means they get in and get out relatively quickly, a matter of weeks. Or they can be opportunists, which are usually residents that have turned over to the dark side and now cause disease. Okay? These guys develop into disease-causing organisms whenever the opportunity arises, or when there's a low amount of other competition, or your immune system is ignoring it. And again, we acquire these from everywhere. Okay, so please keep that in mind. You get them as soon as you come out of your mom's vagina, and then everything you're exposed to, you're going to pick up microorganisms from. And why is it important to have these organisms? Well, first and foremost, they exclude potential pathogen, so take up space. Thank you, I knew it was coming. They can supply nutrition to the host in some cases. They can even help break down nutritional things to make it easier to digest. And then they stimulate the immune response. All right, so let's talk a little bit more about definitions. What is a pathogen? Any organism that inhabits another living organism and can cause disease. And what is its pathogenicity? This is the degree in which it can cause disease, right? So if it has a high pathogenicity, it can cause really serious disease.
Is that also virulence?
So virulence is similar, okay? Except that's from the bacteria's point of view. So what is the bacteria doing to make itself more pathogenic? So, again, pathogen, we just talked about virulence. This is whatever the organism is doing to make itself a better pathogen, okay? And this is usually by producing virulence factors, which can be, really anything. So they can make a capsule, a thicker cell wall, exo enzymes, toxins, what have you. Really, it's anything the organism can do to give itself a growth advantage inside of the patient, which then makes it a better pathogen. Infection, so being colonized by an organism, that could potentially cause disease. And then communicable just means it can be spread from one person to the next. We've already talked about that. Okay, so 17 is a nice summary slide of a generic infection and then subsequent disease. Okay, this isn't exactly true for every pathogen, but it's pretty close. Okay, so the first step is finding a portal of entry. What does this mean? Yeah, finding a way to get in. Right. Most organisms need to get into the body to cause disease. Okay. So they find a way to get in. They can use the skin, the GI tract, the respiratory system, the urogenital system. Any area that is exposed to the environment is a potential portal of entry. Okay? Next, we have the organism attaching. Why does the organism want to attach? Maybe. Kind of hide itself. But usually it likes to attach so that it can get food and nutrients. Right? So it's going to stick itself wherever the nutrients is present. How can it attach itself? There's two ways. For bacteria, at least. Fimbriae, which are small hair-like structures. And capsules, okay? Glycocalyxes. Slimy layers around the organism and merge together with other bacteria, creating a biofilm. Now once they attach, they then need to survive the host defenses. You want to do this because if you don't, the defenses will kill you, okay? The most common things to survive against are going to be phagocytosis, being swallowed up by the phagocyte, as well as preventing or killing the immune cells in the area using leukocytogens, okay, or chemicals that kill immune cells. Now, once you've survived the host defenses, you then can cause disease. What is disease? You don't know what disease is by then.
Bacteria themselves destroy each other.
So disease is where you have tissue or cellular destruction. Okay? When this occurs, this is when you get your signs and symptoms, okay? And you start to feel sick. So this is where the organism is destroying your cells and tissues to get nutrients. How can an organism do this? There's two primary ways. Two primary, proteins they can produce. To allow this to occur. Right, toxins and exo enzymes. Okay? So toxins are small molecules that can bind to a cell and then quickly cause it to die. There are two flavors of toxins: exotoxins and endotoxins. What is an exotoxin? So exo means outside. So it's a toxin that is released from a bacteria that is alive. Okay, it's outside of the cell. Are these serious toxins? Yes. Okay. Exotoxins are often the more serious ones. Remember, they can cause organ failure. What about endotoxins?
Oh, those are the ones that can like go back into the cells and wait for a good time. Is that what that is?
No. Yes, endotoxins are only released when a bacteria dies. So bacteria dies, they break apart, the endotoxins release, and then they can cause problems. Okay? How serious are endotoxins? Are they as serious as exotoxins? No. Okay, these guys tend to be a little more milder, and they cause things like fever.
How does it bind to the bacteria?
It's kind of like altruism, right? So if you got a bacteria that's dying, it releases the endotoxin when it falls apart, and now the guys that are still alive get a little bit of a protective benefit. All right, what about an exoenzyme? What is an exoenzyme? So what does exo mean again? Outside. So it's an enzyme outside of the cell. Okay? And they are used to break down cells and tissues. Why do organisms want to break down cells and tissues? Nutrients, right? They get all the goodies from the cells that they kill. All right, so after they cause disease, they then need to exit the host. Why do they do this? So they can go off and infect other individuals. Okay? Now remember, the portal of exit is often the same as the portal of entry, but that is not always